Okay, so in the previous lecture I was explaining how we could create a recombinant DNA, meaning we could cut open a bacterial plasmid, put a piece of DNA into that plasmid that came from a different organism, seal it up, and now we've created what's called a recombinant plasmid. So now our next step is to get a bacteria to actually absorb the plasmid. And this is what we did in class if you were here for our bacteria experiment today. We took the bacteria and we did a method called heat shock. We added calcium chloride, our bacteria, our plasmid, and all of that stuff went in a little vial. And then we put it on ice, and then heat, and then ice again. And that basically beats up the bacteria to weaken the cell walls. And it makes the bacteria what's called competent. That's the name of when a bacteria is ready to absorb a plasmid. Once we did all of that, we gave it some broth, which is sort of like chicken soup. We put it on plates, little petri dishes with agar, which is the stuff that the bacteria eats, and then we put it into an incubator. The next day, what we should see are little dots, and those dots are what are called colonies. A colony is where you basically started with one bacterium, and every time it divides, that doubles. 2, 4, 8, 16, 32, 64, etc., exponentially, and you get just a circle. And that circle, every bacteria in that circle, in that colony, is basically a clone. It's a bacteria that came from the division of the original. So that's the process of how we would now transform our bacteria. That's called transformation. Now, honestly, on the AP exam, they probably wouldn't ask you specific questions about the technique of transforming the bacteria. You should know the definition. Transformation is when DNA gets absorbed by a bacterium. In fact, we talked about transformation. It was the first experiment when they were discovering DNA. It was the R strain and the S strain, and how they boiled the S strain and put it with R, and that was actually transformation in nature. So this is artificially making the bacteria competent to do transformation. But the part that they would really want you to understand on the AP exam, and this is what I would also be testing you on, is this part about screening it. So here's the thing. We made a plasmid. We mixed the bacteria and the DNA in a vial. Great. You know, we've got our vial, and in it we've put our DNA and we've put our bacteria. And we've hopefully shocked them with this little heat shock technique of heating and cooling and all of that. Hopefully our bacteria is going to suck up the plasmid. But the truth of the matter is, there's no guarantee. It's just like if I came in the room with a bunch of candy and I threw it all in the air, some people might get 5 pieces of candy, some people might not get any candy. Now, if I'm a scientist and I'm trying to create a bacteria that does something super important, like make human growth hormone, I'm only interested in the bacteria that actually picked up the plasmid, the ones that were transformed that are now going to be able to make human growth hormone. I don't care about the other ones. I want them to just be dead. They're not helpful to me. So I need a way of screening the bacteria. So there are a few different things that we can do. What we did in the lab is we used an antibiotic resistance gene. All right, so an antibiotic resistance gene, what that means is, here's our plasmid. So remember, our plasmid is a circular piece of bacterial DNA. In the plasmid that we used, we had already inserted, well, I bought the plasmid, but inserted into this plasmid was a gene from jellyfish called GFP, and this gene codes for making a protein that would make the bacteria glow. So our goal in the lab was that we were hoping our bacteria would have absorbed this plasmid, and now they will have that ability to produce this protein and glow in the dark. But in addition to that protein on the plasmid, there's a second gene on the plasmid, which probably was already there naturally. It probably wasn't artificially placed there by scientists, because a lot of times plasmids in bacteria, that's what they do. They have genes for special things. So the plasmid that we used not only had the GFP gene, it additionally had a gene called AMP. And what AMP is it's a gene that codes for ampicillin resistance, meaning that they would not be killed by the antibiotic ampicillin. So this is a great way to screen the bacteria. So what we did is when we put the bacteria on plates, you'll notice that some of the plates that we put the bacteria on, the plates say on them LB. LB stands for Luria broth. That's the food for the bacteria. Luria broth. That's their food. Some of the plates also said AMP. those plates contain the antibiotic ampicillin. It's been added to the agar. So what's that going to do? Well, any bacteria that did not pick up the plasmid will be killed by the antibiotic. And those that did pick up our plasmid, those that were transformed, they will be able to grow despite the presence of this antibiotic because they are ampicillin resistant. So the colonies should grow on here should only be the glowing bacteria. We shouldn't see any bacteria that don't have the ability to glow because they will be killed by the antibiotic ampicillin. So this is one possibility, is that we use a plasmid that in addition to whatever gene we wanted to insert, also contains a gene for antibiotic resistance. It could be, sometimes there's a gene called CAN, which is for kanamycin resistance. You could have penicillin resistance. You could have streptomycin resistance. There's lots of different antibiotic resistance genes that we could use. Okay, a second option is that we could use a gene that makes the colony a different color. For example, technically, if we just grew bacteria and we added a plasmid, let's just say that the plasmid contains a gene that makes the bacteria grow blue. And it also contains a gene, let's say this was a gene for human growth hormone. So we're trying to make bacteria that produce human growth hormone. Well, what we can do is when we grow these, so we do our transformation experiment, we put our bacteria on our plate of agar, LB agar, which is our food. What's going to happen is nothing will kill the bacteria this time. We didn't add an antibiotic, but since we added a gene that will make the bacteria blue, then we'll know that the colonies that come out blue contain the plasmid, and the ones that just grow regular white don't contain our plasmid. So if we can include a gene that makes the bacteria a different color, we could physically see and then use a loop and take out the ones that picked up our plasmid. So this is a second way that we could screen our bacteria. We could include in the plasmid a gene that makes the bacteria a different color so we could even see those bacteria. A final thing we can do is we can include a gene that allows the bacteria to digest something special. And a great example of this is lactose. So regular E. coli may not have the ability to digest lactose. So let's say that I'm going to create an E. coli that carries human growth hormone. So here's my my human growth hormone gene here on the plasmid. And then additionally on this plasmid, we're going to add the lac gene. And the lac gene is a gene to digest lactose, which is a disaccharide. So now all we have to do is when we grow the bacteria, the way we're going to screen them is we're not going to grow them on LB agar because LB, luria broth, is food. We're going to grow them on what's called minimal media, meaning it's agar, but it's literally, it has no food in it. It's just like a surface. And add to that the only food source for the bacteria, lactose. So now the bacteria that picked up our plasmid will be able to grow because they can eat lactose, they can digest it, but the ones that do not have the plasmid, they'll starve to death because there's no food that they're able to eat. So these are some ways that we can screen the bacteria once we've created them. Now, I made a little practice sheet here, and I need to explain it because it's not very clear what I'm asking you for. So these are just some examples of different bacteria that we could screen. So the first one is we have a plasmid that contains penicillin. Not penicillin, I'm sorry, a plasmid for penicillin resistance. So we have 3 plates. A plate that just has agar, a plate that has agar plus penicillin, and we have a plate that has agar plus a different antibiotic, streptomycin. And the question is, what bacteria will grow? Well, in just plain agar, since there's nothing to screen them, all bacteria will grow. In other words, the ones that picked up our special plasmid for penicillin would grow, but so would the ones that didn't. So you would have no screening at all. You'd have just like this, what we call like a carpet of bacteria. It would just be completely covered in them. Now, second plate, we have agar, but we've added to it the antibiotic penicillin. So in this plate, only the bacteria that picked up the plasmid will grow. The ones that did not pick up the plasmid will be killed by the penicillin. So this one would be a good way to screen these bacteria. And this third one, we added a different antibiotic, streptomycin. In this plate, nothing will grow because our bacteria does not contain any kind of plasmid to make it resistant to streptomycin. So streptomycin, this different antibiotic, would kill everything. So nothing would grow on that plate. All right, so that's our first one. Second one, bacteria with a plasmid that makes them turn blue in the presence of lactose. Again, this is not explained very well. So we have a bacteria, and if they pick up the plasmid, then in the presence of lactose, they'll turn blue. So we have two plates here. The first one just has agar. The second one has agar plus lactose added to the agar. And we can assume this is LB agar, meaning it has plenty of food. So the bacteria don't have to be able to digest lactose. In fact, it says that this plasmid, it doesn't allow them to digest lactose, it makes them turn blue in lactose. So in our first plate of just agar, we have no way of screening our bacteria. So in our first plate, again, we don't have anything going on to screen them, so everything will grow. Not only will all of them grow, but they'll all look alike. So you'll just have a bunch of bacteria growing. Some picked up our plasmid, some didn't pick up our plasmid. In our second plate, everything will still grow because we don't have anything here. Lactose doesn't kill bacteria. But in this plate, we'll be able to tell which ones picked up our plasmid because the plasmid makes them turn blue when lactose is present. So you'll have blue colonies, and you'll know that the ones that are blue are the ones that picked up the plasmid, and the ones that are just plain old white are the ones that didn't. So in our second experiment, we used a plasmid that made them a different color in the presence of lactose. Therefore, if we grow them in lactose, we'll be able to tell which bacteria picked up the plasmid and which ones didn't. The last one, we have bacteria with a lactose digestion plasmid. So this time, if they pick up the plasmid, they will gain the ability to digest lactose. So here we have 3 plates. The first one is just regular LB agar. Well, just like in the other 2 experiments, everything will grow because there's nothing there to screen them. So the ones that can digest lactose will grow. The ones that cannot digest lactose will grow. This second one is supposed to be LB agar with lactose added. Now, this is kind of a trick question because here's the thing. The bacteria that have the ability to digest lactose will eat the lactose. But since this agar, and like I said, I meant to put LB here, also contains luria broth, which is food that any bacteria can digest, we're still not going to screen this. Everything will grow. In other words, the ones that can digest lactose will digest the lactose, and the ones that can't will just eat the other food. It'll be sort of like if you're lactose intolerant, but there's also candy. You don't need the lactose. You can eat the candy instead. The last one is agar that contains lactose as the only food. And that one would screen them. So if there's no luria broth in the agar and the only food available is the lactose, then the only bacteria that would grow in this plate would be the ones that picked up the plasmid. So these are different examples of ways that we could screen our bacteria. Now, why would we want to do this? There's lots of reasons. We can make all kinds of things from it. Bacteria that clean up oil spills. I mentioned several things. Bacteria that make human growth hormone. But we can also do this. Here's the thing. Remember that our cells, our DNA, when we make proteins, our DNA has introns, pieces of DNA that don't code for anything. So if we just take the gene for human insulin off of a DNA in a human, and we try to put this in a bacteria, the bacteria won't be able to read it because it contains introns. So the way that we do it is we use a special enzyme called reverse transcriptase, which happens to be an enzyme that HIV viruses have. Reverse transcriptase turns RNA into DNA. So why would we want to do that? Because what we can do is let protein synthesis start in a eukaryotic organism. Let the DNA make RNA. Let the mRNA go through the processing where all the introns are cut out. Now you have an mRNA with no introns. Now add reverse transcriptase, and we can make a DNA segment with no introns and then put that into the bacteria. So it's a little more complicated when we want to put a gene from a eukaryotic organism into a bacteria. We have to process the mRNA first, so the introns all get cut out, then use reverse transcriptase and to go backwards and make what we call cDNA, complementary DNA. And that DNA will now have no introns, and the bacteria would be able to read it.